So I think there's also a meeting going on right now. Okay, so as you may be aware, we are actually in the next section. So in terms of where we are, we have wrapped up chem. We are now moving into biology. Okay, so I set up a biology folder. Now I actually set up also a course outline. So this is the biology folder. Okay, so right today we're just going to introduce the cell. And then if you want to start looking at this worksheet, so a worksheet on the cell, parts of the cell, and these are the answers below it. If you haven't done the quiz, the online quiz, you can work on that. I did set up an online Google Doc, which just remembered right now, some of you asked to get permission to it. I don't think I actually allowed people to see it. So let me actually open this up so you can see what I mean. So this is the Google Doc that I created for grade 10 course outline on the, so I divided into the cell and cell parts. Now I believe I have to change the permission settings on this. Should be able to view. Just so anyone with the link can view this. Copy link. Done. So I'll send this to you so that you have access to this. So this is the Google Doc with questions on the cell. So you could see here, parts of the cell. Then we'll move on to cell division, and then we'll look at cell transport. Okay. So today we're just going to introduce the cell. All right. Any questions about that? About where we're heading? We're good. Okay. So let's introduce the cell. And if you have any questions, just jump in on the chat. All right. So here we go. All right. So we're going to introduce the cell. Now I think you might have done a little bit about the cell probably in elementary school. I'm not sure exactly when. So what we're going to do today is we're just going to introduce cell theory and parts of the cell. Try to keep this as simple as possible. So we know cells are really, really tiny. And we know that they make up living things. They could have small, very small genomes, which what a genome is the instructions to build the cell, the instructions to build the organism itself. So for example, here is an example of a bacterium. You can see it has about less than 200 genes. To put that in context, we have about 20,000 genes in our genome. But whether you're, a large organism like a human or you're a tiny little cell, you're going to need a certain amount of genetic information to be able to, carry out certain things like growth, reproduction, how to get energy. And that's what we're going to kind of introduce today. So here's an idea of scale. So you can see on the right, humans, all the way on the left is the atom. You could see the virus is over here, and then we have the cells, typically in about this range. So this is actually a logarithmic scale. Every step is a factor of 10. Kind of reminds me of the pH scale. So, if you were to look at this scale, it looks like cells are about 10 to maybe 100 to 1,000 times bigger than a typical virus. So, the cell. One of the first people to actually look at a cell under the microscope was a gentleman by the name of Robert Hooke. So he looked at something like this, and he called them cells because it kind of reminded him not of jail cells, but of where a monk used to actually reside and work out of. So they call them cells. So here's actually a piece of cork. You can see the little rectangular cells that make up the cork. The cell theory is a description of, basically the role of cells in life. So for instance, it says that, according to the cell theory, that all living things have to be made up of at least one cell. So if I go back to, for instance, this image, the thing on the left and the thing on the right, these are living things. And these are things that would be made of at least one cell. So on the right is actually a bacterium. I think the one on the right is Mycoplasma laboratorium. It was actually created in the laboratory, if I remember correctly. I could be wrong. That could be actually the first one, Carcinella rudi. But it is definitely a bacterium. And that means it's made up of only one cell. Whereas the organism on the left is some sort of looks like some sort of an insect. It's made up of obviously more than one cell. But the cell theory states that if it's living, whether it's made of one cell or many cells, it's living because it's made up of cells. All right. So if it's alive, it has to be made up of cells. So then what's a virus? So like something like COVID-19. Well, viruses are not made up of cells. The scale says that viruses are much smaller than cells. So for example, here's a virus, and here's the smallest bacteria. So you see viruses are things that would actually even attack bacteria. So they're not made of cells. They're smaller than cells. So then we would probably have to say that it's not a living thing based on this classification, right? So if all living things are made up of at least one cell, viruses are not living things. Although they have things in common with living things, like they can reproduce, but there are some distinct differences. The large thing that this bacterium is attached to is, the virus is attached to is a bacterium, E. coli. This organism here is a paramecium. These are what they both have in common, E. coli and a paramecium, is they're both, unicellular and both made of one cell. But that's the definition of life. Made up of at least one cell, then you are considered a living thing. This is a cell dividing, okay? Because the second part of the cell, or the third part of the cell theory actually says that all cells come from pre-existing cells. In other words, you, which are made up of trillions of cells, came from, one cell called a zygote, which came from cells from mommy and daddy, and then mommy and daddy came from cells from their mommy and daddy, and that's true of humans. It's true of bacteria. This paramecium would have come from a pre-existing cell. This E. coli would have come from a pre-existing cell. This is a neuron. This is a nerve cell. It would have come from a pre-existing cell that gave rise to it. So all cells come from previous cells. I guess we could say the only exception would be the first cell, right? The first cell would have to come from something that wasn't pre-existing. That's a topic for a different date. Okay, the second part of the cell theory says that all cells are the basic unit of structure and function. So what exactly does that mean? So what it means is if you can think of a function, it's going to be the responsibility of a particular cell to carry out that function. So for instance, these are nerve cells. So think about what the nervous system does. Even something like thinking, right? You have a math problem, you're going to solve it. This requires a particular function that has to be carried out by particular cells. So thinking would be a function carried out by the nervous system. Movement would be a function that requires specific cells, muscle cells, to contract and allow you to move. So functions that are carried out by your body are conducted by very specific cells. So, for instance, we have to move oxygen around our body. So, what cell carries out that function? Does anyone know what cell allows you to move oxygen through the body? Anyone want to guess? Nobody? Maybe too shy? All right, it's a red blood cell. Oh, so someone had it. I think someone came up. Well, let's see what you have to say. Yeah, very good. Red blood cell. Yep, excellent. So red blood cell, and we'll study the red blood cell when we look at something called specialized cells. We'll talk more about red blood cell later on. Okay, so that's the cell theory, right? The three parts. If it's living, it's made up of cells. So viruses are not living because they're not made of cells. All cells come from previous cells, right? And you came from something called the zygote, came from sperm and egg cell, right? And then the cells carry out functions in your body. So we, talked about the example of like the nervous system, nerve cells. Sorry. Okay. All right. So here are things that cells have to be able to do to survive. They have to be able to do the following things. One is you have to be able to control what goes in and out of the cell. You have to be able to move things through the cell, whether that's into the cell or out of the cell or even within the cell. You have to be able to make energy. You have to be able to make proteins. That's actually very, very important, the ability to make proteins. So for instance, the red blood cell makes something called hemoglobin, which allows it to carry oxygen, right? Very, very important function of cells. All cells have to be able to make proteins. They have to be able to get rid of waste, and they have to be able to talk to each other or talk to some other cell. So here would be an example of that, going back to these two neurons, these two nerve cells. You notice that it looks like they're kind of connected to each other because they actually communicate with one another. It's a very, very important concept that cells must be able to talk to each other. Give you like one example off the top of my head. When you eat something that's sweet, that sugar rush is actually detected by cells in your pancreas called beta cells, and they then release something called insulin so that your muscle cells can absorb the sugar. So that's a neat example of communication, right? Your beta cells in your pancreas communicate to your muscle cells through a chemical called insulin, and that tells them to absorb sugar. So these are things that cells must be able to do to survive and be successful. The other thing is reproduce. So when you saw that here, it's actually part of the cell theory, cells coming from pre-existing cells. So here's a cell actually splitting in two, it's dividing. That's actually also very important function of cells. Okay, so we talked about cells, they're really, really small. We'll talk more about why they need to be small at a later date. There's actually a reason why they can't be too big. There's a limit to their size, but we'll come back to that later, okay? We'll talk about this slide when we talk about cell size, okay? We're not gonna talk about that today. We could divide cells into two categories. There are cells that basically are called prokaryotic cells. These are things like bacteria. So for instance, like this is E. coli. These are cells that do not have what's called a nucleus, all right? So when we look at the parts of the cell, the nucleus is a very, very, very large and identifiable part, which I'll show you in a minute. Bacteria do not have this, okay? So The other type of cell, which are called the eukaryotic cells. So here's an example of a prokaryotic cell. All right. Um, the eukaryotic cell does have a nucleus, and you can see that in this slide. Okay, so the nucleus is actually a really important organelle. So it's we'll call that a little body part of the cell, an organelle. And you can see it right here. So this is my nucleus. Okay, just circling it. And its role is to protect a very valuable molecule that I'm sure you've heard of called DNA. All right. And this is important because the DNA is the set of instructions that allow a cell to basically carry out these very important functions like controlling movement, capturing energy, making proteins, communicating, and getting rid of waste. I like to think of the DNA as like the software of a computer that allows, you have your phone or your tablet or computer, and you have all the software and all the apps that do specific things. Think of that as your DNA, okay? Without that DNA, without software on your phone, or your tablet or your computer, it would do nothing, right? Because the DNA is like the programming. It allows the cells to execute very, very specific functions. So that's a good way to think of it, all right? And it's so valuable that the DNA has to be protected. And that's the role of the nucleus. The nucleus protects the DNA because the software is so important. And you know that if your software ever glitches or becomes corrupt, that could be a major, major problem for your phone or for your tablet or your computer. So it's very, very, very important that that DNA is protected. But this goes back to the difference between eukaryotes and prokaryotes. See, prokaryotes do not have a nucleus, so there's no protection around the DNA. We have regions of where the where the DNA is, and that's in blue here. Maybe multiple regions, but there's no layer around it that protects it. So their DNA is a bit more vulnerable than it is in eukaryotic cells. Okay, so the major difference between those two types of cells. So the prokaryotic cells again are bacteria, and everything else are made up of eukaryotic cells. So that would be us, insects, raccoons, fish, plants. They're all eukaryotic cells. Okay. All right, so let's look at some of these parts of a cell, all right? Again, and they're all related to the function of the cell. So the first part is, and this is what all cells have in common, is they all have something called the cell membrane or what's called a plasma membrane. That's the outer part of the cell. So both eukaryotes and prokaryotes have a cell or plasma membrane. Okay, you can use those two terms interchangeably. And they actually, their job is to control what goes in and out of the cell, because a cell wants to be able to have some sort of a decision on as to what can go in and what can go out of the cell, right? So you can imagine that the cell, maybe it absorbs something very valuable, for instance, maybe like glucose, sugar, right? Like imagine a molecule of glucose that is out here. So we'll call that glucose. Glucose is sugar. And the cell needs it, maybe to make energy, okay? Well, that molecule of glucose has to get into the cell and stay in the cell. In other words, the cell has to be able to bring it in without losing it. And that's the role of the plasma membrane. It has to be able to control what goes in and then what goes out. It's very, very important. The plasma membrane also allows cells to communicate with each other. So, for instance, we have special cells in our body called white blood cells, which I'll draw in black here. So I can't draw it in white because then you won't see it. And what they do is, I'll call this a white blood cell, WBC, and their job is to tell whether or not a cell belongs there or not. Because what their job is to protect you. So what a white blood cell does is it goes through your body and it looks for things that are dangerous, that shouldn't be there. And how does it do this? Well, it has to be able to tell whether or not a cell is supposed to be there, whether it's a normal cell or whether it's an invader, whether it's foreign or dangerous. And how does it know that? Well, it knows that because on the surface of the cell membrane, now, if I look at this picture, it kind of looks like the surface is nice and smooth. It ain't smooth. There are like bumps on it. There's like features on the surface of the cell that allow other cells to recognize it. Okay, and this is important for your immune system to be able to recognize cells to know whether or not they belong. So, for instance, you might know, like transfusion, like you just can't give blood to anybody. There's a specific set of rules for giving blood. Why is that? Because your immune system might look at certain blood that you get from somebody else and might say, oh, this does not look like it should be here. And then your white blood cell attacks it. So the cell membrane is not just important in controlling what goes in and out, but it's also important in communication, being able to tell other cells, hey, I'm okay, I should be here. Or telling white blood cells, uh-oh, we have a problem. We've got to get rid of you. Okay. There's a massive term here at the bottom of the slide called hypercholesterolemia. Just a quick word in biology. Whenever you see the prefix hyper means a lot. Okay. So what is this hypercholesterolemia? Okay, well, it sounds like it's an issue with a lot of cholesterol, right? A lot of cholesterol. Now, why am I putting that term on this slide? Because there are special cells in your liver, and their role is, I'm pretty sure it's in the liver, yeah, it's in the liver. Their role is to absorb, remove cholesterol so that the amount of cholesterol is not too high. So what these cells do is they take the cholesterol and let's say I'm going to call it C, and they bring it into the cell so that the cell then breaks it down, for instance, and gets rid of it. Problem with hypercholesterolemia is there's a defect in the surface of the cell that allows the cell to bring in the cholesterol. So if the cell's job is to remove the cholesterol, but we have a defect, and the defect is in the cell membrane of the cell, and it can't take in the cholesterol, then what happens is you have a patient that has too much cholesterol running through their body. And that's a problem because a lot of cholesterol is linked to heart disease and issues as such. So that's just an example, okay? All right, so all cells have a cell membrane, that's the outer part. All cells also have what's called the cytoplasm. That's the inner part. So in other words, I like to think of it as like the ocean of the cell. So that would be all of this stuff in here. This is like the jelly of the cell. This is the cytoplasm. It's mostly made out of water, but there are other things in there, like water, salts, and maybe some sugar in there. Okay, and it's basically where the things that are called the organelles So the organelles are like the organs of a cell, okay? These are the little like body parts of the cell. So the organelles are the things, the little body parts of the cell that would actually be floating around in the cytoplasm, like the ocean of the cell, okay? So all cells have a cell membrane, all cells have cytoplasm. Not all cells have a nucleus though, because prokaryotes don't have a nucleus. Here's a cool picture of a cell that's been frozen. So you could see the cell membrane, right? The outer boundary of the cell. You see that here. You could see the nucleus right here. So obviously this is a eukaryotic cell. And then in between is the cytoplasm. But you can see that there's stuff in the cytoplasm. Those are the organelles, body parts of the cell. All right, so we talked about what the nucleus does. The nucleus has a job. Its job is to protect the DNA. There is an interesting genetic disease that is due to a defect in the nuclear. Because if I look at the nucleus itself actually has a boundary. Okay, and that nuclear boundary, you can see that there's like an outline to the nucleus. That nuclear boundary is called the nuclear membrane. So just like the cell has a cell membrane, the nucleus has a nuclear membrane. If I change, see if I can change this color. So it's called the nuclear membrane. And there's an interesting defect called progeria, which actually is due to a defect in the nuclear membrane that actually causes really rapid aging. So these unfortunate individuals that are born with progeria, which again comes down to a nuclear membrane problem. You can see the top right is an image of a child, an image of normal nuclear membrane, and in the bottom right is abnormal nuclear membrane. And this abnormality leads to a condition called progeria. And you can just tell there's something not quite right here with this child. This is going to cause rapid aging. I'm not really sure exactly how long these children live for, but I don't think they live past the age of 15 to 20 years, something like that. So it's very, very catastrophic problem. Okay, so let's talk about your DNA. So, I should mention one more thing. Going back to the nucleus. So the nucleus. So this is the nucleus. You can see it's highlighted here. There's the nuclear membrane, also called the nuclear envelope. All right. Some of these things have multiple names. You can see that there's these little like spaghetti-like structures. That's actually your DNA. All right. Now your DNA, although I don't see the word DNA on the screen because their DNA is actually attached to these things called proteins, especially in this slide here. And the DNA and protein form something called chromatin, which you can see here as these little spaghetti-like structures. But basically, the little strands are, pieces of DNA. The instructions we talked about, the software of the cell. I can also see that the nucleus has these pores, and the reason why it has the pores would suggest that things must be able to go in and out of the nucleus, right? Must be able to go. Otherwise, why would they have these little doorways into the nucleus? And the reason for that, and you'll learn more about this in like grade 11 and grade 12 biology, is the cell needs access to the DNA. So if you think of the DNA as like the software, this is software typically on how to build some sort of a protein. So let's say the protein is something like hemoglobin, or maybe insulin. Okay, the DNA. Think of it like a recipe on how to make these proteins. Now, the recipe is in the nucleus, but the actual proteins are made outside the nucleus. So if the information is in the nucleus, but we make it outside the nucleus, then what has to happen is the information has to go from inside the nucleus to outside the nucleus. Okay. It would be kind of like this example. Imagine that it's not my example. So I'm not going to claim that I can come up with this. Imagine that the nucleus is like a library. Okay. Imagine this is like a library, and the little spaghetti strands are books or articles, and they contain recipes. Now, you go to the library and you want to read a recipe on how to make a pizza. Now, you don't make the pizza in the library. It's not going to work. So what you do is you go to the library and you make a copy of the recipe, and then you take that copy outside the library and you take it home and you make the pizza. And the cells do the same thing. So in the nucleus is the information on how to do things like make proteins, but that information is actually copied in some way, and it leaves the nucleus, and then it's used to make that information is then used to make a protein. I'll show you exactly where that actual information goes in a minute, okay? But I do want to show you this. So this is actually a slide of DNA, right? So this is where you would find the instructions on how to make things like insulin, hemoglobin. Um, also in here are the instructions that determine things like hair color, skin color, and so on and so forth. Here's a really interesting picture of what happens when there's a glitch in the software. You can see there's something unusual about this fruit fly. Can anyone guess what's unusual about the head of this fruit fly? Anyone want to take a guess? There's something really unusual about this fruit fly. So this is a neat example of where there's a glitch, there's a problem with the software, there's a problem with the DNA. Because not only does the DNA tell you what to make and how to make it, but it also tells you where to put things. Like, for example, in your DNA are instructions that say, your eyes go in a certain spot, your arms go in a certain spot, your head is here, and your shoulders are here, right? Those are all laid out in your DNA. So can anyone spot the mistake here? Right here. So you see this structure right here? These are legs. So you can see here, we have a problem here. We have legs growing out of the head of a fruit fly. It's a neat example of where a mistake is occurring in the programming, the software, that's obviously causing an interesting what's called mutation here, okay? This is what's called a chromosome. So a chromosome is when your chromatin that looks like a little spaghetti, it actually thickens up, it condenses, and it forms these things called chromosomes. So in that chromosome would be the DNA. We'll talk more about chromosomes when we talk about cell division, okay? So we'll come back to chromosomes. Now, I mentioned that the nucleus has your DNA, the set of instructions on how to make things like proteins. That information is protected by the nucleus, but it has to leave because, like I said, you can't make a pizza in the library, so you can take the information out. So where does it go? So where it goes are two possibilities. One is it goes, so if I look at this picture here, you'll notice that there is, here's the nucleus again. And then it looks like there's like a maze-like structure that's attached to it. So this maze-like structure is called the rough ER. Okay. It's called the rough endoplasmic reticulum. You'll notice that it's got these little black dots. Okay. Now, some of it looks like it does not have black dots. So if it doesn't have the black dots, it's actually called smooth ER. So kind of like, you know, when someone has shaved, it looks smooth. So you can see the difference between the rough ER and the smooth ER. So here's actually an image of that. So here's the rough ER. You can see the little black dots. You can see the little black dots. And then here is the smooth ER, where I don't see any of those black dots. The black dots are actually structures called ribosomes. Okay. Now, the ribosomes, if I go back to my pizza analogy, think of ribosomes like chefs or cooks. Okay. What a ribosome does is a ribosome will read the copy of that information that comes out of the nucleus and convert it into some sort of a protein. So remember I said to you that the nucleus contains the information. It's valuable, protect it. You can't take the information out, but what you can do is you can make a copy of the information, and that copy could leave. So you can go to the library, you can't take the book out, but you can make a copy of the book and take it out. That information is instructions on how it's not about making a pizza, it's about making proteins. And where it goes is it goes to these chefs, these little ribosomes. And what they do is they read the recipe and they make that recipe into some sort of a protein, like hemoglobin or insulin, two examples I've been using. Now, the ribosomes can be either free, which means they're literally just floating around the cell, or they could be bound, they could be attached. So in this image here, you can see my ribosomes in black are attached. When they're attached to the ER, they're called rough ER, okay? But whether they're bound like here or whether they're free, they have the same job. Their job is to take the instructions that come from the nucleus and convert it into some sort of a protein. Okay. The big difference between the two, though, is so like what happens if a ribosome that is free reads the recipe compared to a ribosome that's bound. So they both do the same thing. They both read the recipe and they both convert it into a protein. The difference between the two is what they do after they've made the protein. So if a free ribosome makes a protein by reading the instructions, that protein will stay in the cell. It doesn't leave. Like hemoglobin doesn't leave the red blood cell. So that would be another example of one we could use, hemoglobin. Okay, here's another protein. This is a protein that actually its job is to make energy. So if a free ribosome makes some protein, that protein does not leave the cell. It stays in the cell. So if this is my cell, okay, that protein would stay in the cell. It does not leave. But if a bound ribosome makes it, so that would be a ribosome that is attached to the ER, then that ribosome produces a protein that does leave the cell. For instance, if we go back to the example with insulin, uh, the pancreas makes it, but it leaves the pancreas and it goes to your brain, it goes to your muscle cells. So obviously it has to leave the cell. Um, so those are the two possibilities. Either the protein stays in the cell or it leaves the cell. If it stays in the cell, it's because it was made by the free ribosome. If it's leaving the cell, it was made by a bound ribosome or basically the rough ER, okay? And again, if I go back to this picture, you can actually see the two types in this slide. So you can see the little black dots. Those are the free ribosomes. So you can see them right here, the free ones. And then the ones that are attached to my membrane, my ER, those are the bound uh, ribosomes, which make up the rough ER. Any questions? Because I know this is quite lengthy, and there's quite a bit here. Any questions at this point? Because I think I'm going to do one more example, then end it, and then next time we'll do like part 2. Okay, so we'll split this up. Otherwise, it's just quite a bit of information. It's quite overwhelming otherwise. All right, so let's look at one more body part of a cell, one more organelle. So going back to this slide, you'll notice that not all of the ER has the ribosomes. So part of it looks smooth. So what does the smooth ER, what does it do? Okay. It has a different function. Whoa, skipped over it. Oh, right here. No, where is it? There we go. Okay, yeah, smooth ER. So the smooth ER has a different role. It doesn't do the same thing that the rough ER does. So it doesn't make protein. It actually makes things called fats. And it's also involved in breaking down. That's what this term means, detoxifying. So detoxifying means to break down things that are toxic or poisonous. So what's really interesting about the smooth ER is that what would happen to someone who's an addict is, for instance, let's say someone who's has an alcohol addiction. What tends to happen, and this is true of any drug, is that as you use a drug more often, it will take more of the drug to have the same effect. So in other words, if you have someone who doesn't drink alcohol and they have a little bit of alcohol, they'll have a noticeable effect. Whereas if you have someone who drinks lots of alcohol and they have a little bit of alcohol, they won't even notice it. So what's going on there? Why is it that someone who's an addict needs to have more of the drug to see the effect? Well, this is actually a really important concept in biology, is that there is your body does adapt to things. What happens then is an addict will actually start producing more smooth ER. And because they produce more smooth ER, they actually become better at breaking down toxic substances like alcohols or drugs. So that means then that for them to have the same effect from that alcohol or drug compared to someone who doesn't use these things, they would need to take in more of that alcohol or drug because their body is simply adapted to become better at breaking down these toxic substances. So it's an interesting little fact there. Actually, let's just mention one more thing and then we'll wrap up, because this is actually related to what we were just talking about previously to the smooth ER. So we just distinguished the difference between the smooth ER and the rough ER. So going back to the rough ER then, we said that the rough ER is, it's similar, like they have a similar job to what the free ribosomes do. They make protein. The difference, though, is the rough ER would send that protein out of the cell, right? So any protein that's made in with the free ribosomes, we said, stays in the cell, but proteins made by the rough ER would actually leave the cell. Okay, so what actually happens is for them to leave the cell, they have to go to another organelle, which I like to call the purulator or the Canada post of the cell. This is called the Golgi complex. Here's what it looks like under the microscope. So if I zoom in on the Golgi complex, its role is to take in things from the rough ER. So the rough ER would send material to the Golgi, and these are the little packages. So, you know, if you're going to send a package to Canada Post, you put the package in some sort of a container. Sorry, let's say you wanted to ship something, you know, to Barrie, Ontario. What you're going to do is you're going to put it in a package. That's what I meant to say. You put it in a package, right? So maybe we want to ship, like, for instance, insulin. So maybe in here we have, in this little red package, there is insulin. I'm going to call I. That insulin would be in a package. That package is called a vesicle. That's the name of the package. And it's received by the Golgi complex. This is my Canada Post, my purulator. So the rough ER sends stuff to the Golgi complex. The Golgi complex then will ship it. That's its job. Its job is then to ship it. Now, where does it ship it? It ships it to the cell membrane, because if you're going to leave the cell, right? Remember, going back to the role of the cell membrane, it has to, the cell membrane is what actually controls what goes in and out of the cell. So the Golgi complex will send something to the cell membrane, which then will, you know, get rid of it so that it could go to some other cell or some other part of your body. Okay, I think that's enough. I think that's quite a bit. Let's divide this, because we got a lot more to cover here. We introduced the cell theory, talked about certain parts of the cell. We still have a few more to talk about, but I think I'm going to stop the record, and then I'll take any questions. All right, hold on. Let me just